Sterilization techniques in the lab depend on what we are going to sterilize. The autoclave is used to sterilize equipment, laboratory instruments, liquid media, and more. In the lab, we use 10% bleach and 70% ethanol as equivalent disinfectants. Both of these disinfectants are used to wipe down surfaces and kill liquid cultures. Autoclave sterilization utilizes high pressure and high temperature steam to kill all living organisms such as fungi, spores, bacteria, and viruses. There are different autoclave settings for autoclaving different materials, such as solids, liquids, other equipment. The autoclave is like a giant pressure cooker since it utilizes steam sterilization. Autoclave prepping. Never put liquids over 50% capacity into the autoclave. Always use a secondary container to contain solids and liquids in case something breaks under the high heat and pressure. Always loosen caps on glass bottles to prevent pressure buildup inside the bottle that may cause it to burst. Always use autoclave indicators to ensure that the autoclave run was successful and that the necessary items have been sterilized. Again, full containers of liquids should not be autoclaved. Always autoclave liquids at 50% capacity to prevent spilling in the autoclave. On the left, we have 3 full 1,000 milliliter Pyrex bottles containing MD solution. We do not autoclave like this. We would split up the 1,000 milliliters into 2 bottles, 500 milliliters in the Pyrex bottle and 500 milliliters in an Erlenmeyer flask. We would cap those flasks with foil and autoclave tape. The Pyrex bottles would have their caps loosened. Autoclave containers. Autoclave containers are usually made of hard plastic, such as polypropylene, that can withstand the high heat and pressure environment. Containers that are not autoclave safe will melt. Autoclave indicators. Autoclave indicators are used to ensure that the autoclave run was successful. In the lab, we often use autoclave tape, which darkens under high heat and pressure. On the left, we see the light color of the autoclave tape before it is run through the autoclave. And on the right, we see the dark stripes that appear after the tape has been run through the autoclave. In order to load the autoclave, we have to make sure that we have provided autoclave tape, a secondary container. We have made sure that all the lids are loosened, the glass containers are not over 50% full, and that our bottles are labeled with initials, the date, and the media type. Now we are ready to load our supplies into the autoclave. Once the autoclave is loaded, we close the door firmly and wait for the pressure seal to shut. Then we hit run. It is always a good idea to wait a few minutes to ensure the autoclave is running and displaying the green run light. Here is a short video of the noises the autoclave will start to make. The autoclave runtime depends on the setting and the materials being autoclaved. It can vary anywhere from 45 minutes to almost 2 hours. Once the run is complete, the display will read ready, and one can hit open. Allow a couple of minutes for the pressure seal to release and for steam to be released from the autoclave. When unloading the autoclave, allow the steam and heat to escape for a few minutes so the items can cool down. Always wear heat-proof gloves over your latex gloves when handling the equipment. If needed, use a cart to transport the hot equipment. When finished with the autoclave, leave the door cracked open so moisture does not accumulate inside. Here's an image of the autoclave door cracked open. A successful autoclave run has autoclave tape which is darkened, minimal spillage of the liquids, and no breakage of the items being autoclaved. Now that the autoclave has finished, the media can be consolidated back into the 1000 milliliter Pyrex bottles. Autoclave safety hazards. The autoclave is a very dangerous piece of equipment due to the intense pressure that builds up inside. Autoclaves should always be treated with caution. Furthermore, one should always use heat gloves when taking things out of the autoclave. And also be wary of the steam exiting the autoclave when opening the door. Lastly, make sure that the materials you are autoclaving are safe to be autoclaved. For example, not all plastics can withstand the heat of the autoclave. If you are unsure that the materials you are trying to autoclave are autoclave safe, it is best to check in with the TA or the lab manager. Now on to 70% ethanol and 10% bleach. These disinfectants effectively kill pathogens such as bacteria and microbes. These solutions are used to wipe down surfaces such as lab benches, equipment, and gloves. Both of these disinfectants are also used to be poured into liquid cultures to kill active microorganisms. 10% bleach is often used to inactivate liquid cultures. In some cases, we would use 9 parts of solution plus 1 part 100% bleach to make it a 10% bleach solution. In the CureSits lab, bleach lives underneath the sink and also in squirt bottles found on the lab bench. Why do we use 70% ethanol as a disinfectant? At concentrations above 70%, the effectiveness of the disinfectant decreases. 70% ethanol is used as a disinfectant in the lab, while 90% ethanol is used as a solvent. Be sure to read the labels on squirt bottles.